Welcome to part 2 of this series on antimicrobial drugs. In this video, we'll be focusing on drugs that target cell wall synthesis. Paul Ehrlich was the Nobel Prize-winning German physician and scientist who is credited with discovering salvarsan, a chemical compound that was the first effective treatment against syphilis. As a result of this work, he was the first person to call the treatment of disease with chemicals chemotherapy. Ehrlich is also credited with popularizing the concept of a magic bullet. He said that in order for a drug or a compound to be a magic bullet, it must target microbial structures that are not shared with mammalian structures. In this video, we're going to look at some magic bullets. Your objectives are to describe the role of bactoprenol, transpeptidase, and autolysins in peptidoglycan synthesis, to describe the structure of peptidoglycan, to explain how beta-lactam antibiotics work, to explain two methods that bacteria use to inactivate beta-lactam antibiotics, to describe 4 methods that scientists use to overcome resistance to beta-lactam antibiotics, and to identify alternatives to beta-lactam antibiotics. Finally, you should be able to name 2 antimicrobacterial antibiotics and briefly explain how they work. This information is likely to be on your exam. Before we move on, let's see if you can answer this question from Chapter 4. Which of the following is not true of the bacterial cell wall? Is it number 1, it provides cell shape? Number 2, it's made up of chitin? Number 3, it's a defining characteristic of most bacteria? Number 4, it protects against osmotic lysis? Or number 5, it anchors flagella? Which one of these is not true of the bacterial cell wall? I hope you said number 2. Remember, the bacterial cell wall is made up of peptidoglycan. Everything else is true. So what I hope you get from this question is a little reminder that the bacterial cell wall is extremely important to the life of a bacterial cell, especially since in the absence of a cell wall, bacterial cells are likely to explode or lyse open due to osmosis. Penicillin refers to 50 chemically related compounds that are all differentiated by their side chain. So what that means is if we were to look at all 50 different penicillins, we would see that they basically have this same structure, every last one of them. It's called the common nucleus. And then the chemistry that's outside of that structure, that common nucleus, is going to be different. Penicillins can be grouped into 3 categories. Natural penicillins have been extracted and purified from molds like Penicillium chrysogenum or Penicillium notatum. These antibiotics have 3 potential disadvantages. Number 1, they're narrow spectrum and only affect one type of bacterium, the gram-positives. They are commonly used to treat staphylococcal, streptococcal, and spirochete infections. Number 2, natural penicillins are not retained in the human body for more than a few hours. For example, when given by an intramuscular injection, penicillin G is excreted in the urine within 3 to 6 hours. The effectiveness of this drug is further reduced by digestive enzymes when taken orally. The third disadvantage is that these drugs are readily inactivated by enzymes called beta-lactamases, which bacteria have evolved so that they can survive a penicillin attack. Beta-lactamases are antibiotic resistance enzymes. Semisynthetic penicillins are natural penicillins that have been extracted from molds, but they were then modified in the laboratory to, number 1, improve retention in the body, Number 2, resist the effects of beta-lactamases. And number 3, extend the spectrum of the antibiotics, so it's effective against gram-positives and also some gram-negatives. Extended spectrum penicillins are a subclass of semi-synthetic penicillins. They're even more effective against gram-negatives because they're resistant to the beta-lactamases that gram-negatives produce specifically, and these drugs may penetrate the outer membrane of the gram-negative cell more readily. Penicillin drugs target peptidoglycan. Remember, this is a unique molecule that we find in bacteria. At this juncture, I think it would be good for us to review what peptidoglycan is and talk about how it's synthesized, as that's going to be important for the rest of this lecture. So peptidoglycan is a disaccharide made up of 2 glucose-derived sugars. So you have N-acetylglucosamine here, which I'm going to call NAG, and then you have N-acetylmuramic acid here, which is called NAM. And notice that the NAG and the NAM are joined together by a glycosidic linkage here in the middle, so we have a true disaccharide. These 2 sugars are repeated in an alternating pattern to form the backbone of peptidoglycan. Now, the backbones of the peptidoglycan are joined together through a group of amino acids that are attached to the NAM molecule at this location. So this is probably carbon number 1, 2, 3, at carbon number 3. On the last slide, I showed you that there's a little linker on the NAM molecules. Well, here's what's attached to that little linker. It's a series of amino acids. And what's important about this is that every NAM molecule has these amino acids attached to it. And then there's a cross-link or a chemical bond that's made between the amino acids of one NAM molecule and the amino acids of another NAM molecule. This interlinks the peptidoglycan backbones together, and it makes the peptidoglycan cell wall of bacteria much stronger. What penicillin is going to do is it's going to disrupt this cross-link between the NAMs, which will weaken the cell wall and make bacterial cells more susceptible to osmotic lysis. To understand how this happens, we need to know how peptidoglycan is synthesized. So you'll need to go to the video that's in your course space in the lecture videos folder that says peptidoglycan synthesis. Did you watch the video on peptidoglycan synthesis? If you didn't, you should go check it out in the lecture videos folder of your course space before moving on. If you did check out the video already, who's awesome? You're awesome. Now let's put everything together. You'll hopefully recognize this as the common nucleus of penicillin. Remember, we said there are over 50 different penicillins, and all of them have this same chemical structure. The business end, that is the part that's actually doing the work for this common nucleus, is this square that's highlighted in yellow. This is called the beta-lactam ring. What the beta-lactam ring does is it binds transpeptidase, and it prevents transpeptidase from cross-linking NAM molecules together. This weakens the cell wall and makes the bacterial cell susceptible to osmotic lysis. The other thing that penicillin does is it stimulates the activity of autolysins. So not only does the cell lose the ability to cross-link its cell wall, but it's also stimulated to break down its own cell wall. This 1-2 punch breaks down or weakens the cell wall to an extent that the cell becomes susceptible to bursting by osmotic lysis. So now let's see what you remember. Autolysins, number 1, are proteins that digest peptidoglycan. Number 2, break down cell walls to allow growth and repair. Number 3, are stimulated by penicillin, or number 4, all of the above? Remember to pause your video and then press play when you've got your answer. I hope you said all of the above. All of the above is the correct answer here. Autolysins do all of these things. Here's another one. Transpeptidase, is it number 1, transports NAG and NAM across the plasma membrane? Number 2, cross-links NAM molecules? Number 3, adds glycosidic linkages between NAG and NAM? Or number 4, breaks down NAM? Number 2. That's correct. Cross-links NAM molecules. Remember, this cross-linking is integral to strengthening the cell wall and helping the bacterial cell resist osmotic lysis. How does penicillin kill bacterial cells? Is it by stimulating autolysins, or binding transpeptidase and blocking cross-linking of NAM, or causing osmotic lysis of bacterial cells, or all of these? Yes, penicillin does all of these. That's correct. Great job.
I want to tell you before we move on that typically your gram-positive cells are most susceptible to penicillin, more so than the gram-negatives. Remember that your gram-positives just have a peptidoglycan shell. So that means that penicillin has pretty clear access, okay, to peptidoglycan and the enzymes that are involved in synthesizing it. For your gram-negatives, on the other hand, remember that the peptidoglycan is sandwiched between the plasma membrane of the cell and the outer membrane. And that outer membrane is an impenetrable barrier to penicillin. The only way that penicillin could possibly get in is through porin proteins, which make a channel through the outer membrane. Unfortunately, penicillins tend to be too big for those porins, and thus they don't pass that outer membrane. The exception are going to be some of your semi-synthetic and extended spectrum drugs, which have been modified so that they can be effective against your gram-negatives. Now, some bacteria are able to make enzymes that will essentially inactivate penicillin. So remember, it's the beta-lactam ring in penicillin that's kind of important. These enzymes will break the beta-lactam ring so that the penicillin is inactive. So in this example, you have penicillinase, which is one of these beta-lactam enzymes that cleaves the beta-lactam ring. And when it acts on penicillin, you get penicilloic acid, which is not effective against bacteria. So what happens is gram-positive cells make these enzymes and then they secrete them into the environment around the cell. And if there's penicillin out there, then the enzyme will break down the penicillin before it gets to the bacterial cell. For your bacteria that are gram-negative, they will synthesize this enzyme, and it basically gets trapped in the space between the outer membrane and the plasma membrane. That way, if any penicillin makes it into the cell, it gets inactivated there. For your gram-positives, the production of these enzymes is induced. That is, the cell detects that there's penicillin around and it starts making the enzyme at that point. For your gram-negatives, it's made constitutively, so at a constant rate. Be certain that you check out the video that I've posted on the mechanism of beta-lactam antibiotics. It will talk about about how penicillin and other beta-lactam antibiotics work, and it also talks about how bacterial cells resist those antibiotics. What you want to pay attention to is the mechanism of activity, so that's how the antibiotics actually inhibit cell wall formation. And you want to pay attention to the methods that bacteria can use to inactivate penicillin, okay, or to prevent penicillin from actually working. I'm not so much interested in you knowing any actual gene names, okay, or enzyme names, but I am interested in you knowing the general mechanisms. So you have these beta-lactamases. These are those beta-lactam antibiotic inhibiting enzymes. They're called beta-lactamases. So there's three ways that we can essentially keep those keep those enzymes, those beta-lactamase enzymes, from destroying penicillin and other beta-lactam antibiotics. One thing that we can do is we can modify penicillin so it's not recognized by those enzymes anymore. Another thing we can do is add an extra chemical to our penicillin drugs to inhibit the enzymes so that the penicillin and other beta-lactamase drugs will work. Third thing we can do is synthesize new antibiotics that still have that beta-lactam ring, but they don't look like penicillin at all. So here we have kind of a summary of how those things happen. The first thing that we can do is make penicillinase resistant penicillins. So a lot of times, again, these are your semi-synthetic penicillins. They've been produced naturally in fungi, but then they've been modified in the lab so that they don't look exactly like penicillin, which will fool the enzymes, okay, so that the enzymes don't recognize these drugs. You also have examples of drugs like your carbaponyms that look kind of like penicillin, but you change one of the atoms in the carbapenem. So you substitute a carbon for a sulfur and add a double bond. And so instead of having penicillin, you have this other class of drugs that looks like penicillin, but it's slightly different. Another thing we can do is add an enzyme inhibitor to the drug. So this is basically what augmentin is. It's amoxicillin, which is a semi-synthetic type of penicillin. Then you add something called clavulanic acid. And clavulanic acid is basically an enzyme inhibitor. So it binds the beta-lactamase enzymes so that they don't work on penicillin. And then another thing that can be done, again, is we can take penicillin and we can really change the way it looks, make another drug, essentially, that still has that beta-lactam ring, but it's not penicillin. So if you look at the little square that's on the right side of the slide at the bottom, figure 20.6a, right under the figure 20.6a label there, you have penicillin in the upper left. Cephalosporins are another class of drugs that are also beta-lactam antibiotics. They have basically the same function as penicillin, but instead of having that 5-membered ring or that 5-sided ring next to the beta-lactam ring, you have a 6-sided ring. So it's slightly different structurally. Your cephalosporins and your penicillins are similar enough chemically that if someone is allergic to penicillins, they're usually allergic to your cephalosporins as well. So it's only a slight change between the two drugs, but it's enough to get around some of your beta-lactam antibiotics. You also have the carbaponyms, which I mentioned already, where we change one of the atoms in the molecule. So if you look here, you'll see that there's a sulfur in penicillin that's been changed to a carbon, okay, in the carbapenem. So this drug is not chemically identical to the penicillins, but it's slightly different, and thus it can avoid some of your beta-lactamase enzymes. And then you have the monobactams lastly. So if you look at the monobactam that's in this figure, you'll see that it's basically just the beta-lactam ring and a little bit of chemistry outside of it. So it looks really different from penicillin, and it would be hard for some of your beta-lactamases to recognize that particular drug, okay, because it looks so different than penicillin. This slide is here just to show you the difference between cephalosporins and penicillins. Again, you have the beta-lactam ring. Everything to the right, to the left of that beta-lactam ring is the same. It's the right side of the structure that's different.
One last thing about penicillin before we're done with this topic. According to the CDC, approximately 10% of all U.S. patients report having had an allergy to penicillin at some point during their lifetime. What does a penicillin allergy look like? Well, while taking penicillin, maybe somebody develops a skin rash or hives. Maybe they have itching or fever or shortness of breath. Could be wheezing. In the most severe cases, they have anaphylaxis, which is a life-threatening allergic reaction. Usually that's accompanied by tightening of the airways and trouble breathing, a weak, rapid pulse, seizures, abdominal cramps, or nausea. In actuality, less than 1% of people are allergic, meaning they produce antibodies against the drug. Are you surprised? It turns out that for most people who react to penicillin, they lose their sensitivity after about 10 years. Still others have reaction to the fillers in the antibiotic rather than the drug itself. In 2019, doctors at Harvard Medical School suggested that if you've been diagnosed with a penicillin allergy, it's worthwhile to get retested when you're healthy. You might be wondering why. Well, if penicillin allergy is on your chart, you will not be prescribed penicillin and possibly any other beta-lactam antibiotic like cephalosporin. The alternatives to penicillin are usually broad-spectrum antibiotics, which have more side effects and an increased risk for C. diff and MRSA infections. Doctors may also opt to prescribe less effective medications. All of these are not ideal. So it's worthwhile to get tested again so that that penicillin allergy can be removed from your chart unless you actually are still allergic. So now let's see what you remember. All of the following are true of natural penicillins except, number 1, they induce osmotic lysis, or is it number 2, they're resistant to beta-lactamases, or number 3, they inhibit cross-linking of NAM molecules, or they have selective toxicity, or they're eliminated from the human body within a few hours. Right, they are resistant to beta-lactamases. That's completely untrue of natural penicillins. Natural penicillins are sensitive to beta-lactamases. Remember, beta-lactamases are enzymes that inactivate or break down penicillin so that it cannot do its job. Here's another one. Beta-lactam antibiotics, like penicillin, inhibit which of the following enzymes? Is that autolysins, bactoprenol, transpeptidase, hexokinase, or peptidoglycan transferase? Right. Transpeptidase is the right answer here. One more. Augmentin contains clavulanic acid. Which of the following is true of clavulanic acid? Is it number 1, it inhibits protein translation by binding the 70S ribosome? Or it changes the spectrum of tetracycline? Or it inhibits peptidoglycan synthesis by binding penicillin binding proteins? Or it inhibits the activity of beta-lactamase enzymes like penicillinase? Or is it that it disrupts the plasma membrane of cells? What do you think? Right, it inhibits the activity of beta-lactamase enzymes like penicillinase. Good job.
So another way that we can get around beta-lactamase enzymes, these enzymes that break down your beta-lactam antibiotics, is we can try to treat bacterial infections with antibiotics that are just not penicillins. They don't have beta-lactam rings at all. And so that's where bacitracin and vancomycin come in. Bacitracin is a pretty easily acquired antibiotic. There are bacitracin ointments, for example, on the first aid aisles at your local drugstore. This was an antibiotic that was isolated from, I believe it was Bacillus subtilis, that was isolated from a young lady named Tracy. So Baci comes from Bacillus, and then Tracin comes from Tracy. In any case, the drug is fairly toxic taken internally, so it's only used topically, and it's effective against gram-positive bacteria like staph and strep, which is, kind of good, because where do you find these types of bacteria but on the skin? Okay. The way that the drug works is it binds bactoprenol. So remember, this is that lipid that sits in the plasma membrane of the cell, and it transports NAG and NAM from the cytoplasm of a bacterium to the cell wall. So this drug binds bactoprenol and prevents it from bringing new NAG and NAM to the cell wall. And you might be wondering, well, why is that important? Well, remember, you have these little enzymes called autolysins that are breaking down the cell wall, and then the cell wall gets repaired and strengthened. If bactoprenol is no longer functioning, every time the autolysins essentially digest part of the cell wall, there's nothing to repair it. Thus, you weaken the cell wall, and the cells become susceptible to osmotic lysis. Vancomycin is a glycopeptide, and you've probably heard of this drug before, because for a long time it was considered a last line of defense drug against MRSA. So remember, MRSA is methicillin-resistant Staphylococcus aureus. It gets that designation because it's resistant to methicillin and several other penicillin-type drugs that would be used for treating Staph aureus infections. So up until probably about 10 years ago, I would say, maybe even a little bit longer, people would get MRSA, and then they could go to a doctor. The doctor would send them either to a hospital or a clinic to get vancomycin administered intravenously, and then the MRSA would be cured. Well, now we have Staph aureus that is resistant to even vancomycin. And so this particular bacterium is called Versa. So instead of MRSA with an M, you have Versa with a V. You also have some strains of Enterococcus that are also vancomycin resistant. So they're VRE or vancomycin resistant Enterococcus. These are the types of bacterial infections that require precautions to be taken when someone's in a hospital. So if you go to visit someone who has VRE or Versa, usually you have to gown up, you have to put on gloves, and you have to be, you're cautioned to be very careful while you're in the room with how many different surfaces and things you touched, because this is a bacterium that you don't want as part of your normal flora, or you don't want to get an infection from one of these bacteria. All right, how does vancomycin work? Well, vancomycin binds the amino acids that are part of NAM. So remember, that's where the cross-link is made. When vancomycin binds these amino acids, it means that transpeptidase and carboxypeptidase can no longer cross-link NAMs, and this leads to weakening of the cell wall. Your important last line of defense drugs against Staph aureus are now Sinercid and linezolid, which we'll talk about later on in this chapter. So we have just a couple more drugs that we want to talk about that inhibit cell wall synthesis. These are specifically antimicrobacterial antibiotics. So these guys are very narrow spectrum. They are not effective against typical gram-positives or negatives. These are only effective against those bacteria that make some kind of mycolic acid and then insert that into their cell wall. So isoniazid is the first one, and it inhibits mycolic acid synthesis. The second drug is ethambutol, which inhibits incorporation of mycolic acid into the cell wall. And both of these drugs are actually used in treating tuberculosis infections. Ethambutol is very often used in addition to other drugs. So if you look at this particular slide, you'll see on the right-hand side that I have some of the drugs that are used for treating TB infections. So you have rifampin, which we'll talk about later. It inhibits RNA synthesis, and it is very often administered with ethambutol together. Okay. You can also have someone on isoniazid, okay, or pyrazinamide. Pyrazinamide, it appears, is active at the plasma membrane of the cell, and it also disrupts some of those enzymes that are part of ATP synthesis. That is the electron transport chain, which you find in the plasma membrane.
Let's try 2 more questions and then wrap this video up. Which of the following is selective against Mycobacterium tuberculosis? Is that chloramphenicol, isoniazid, erythromycin, or vancomycin? Right, isoniazid is the answer here. It targets or inhibits mycolic acid synthesis, and mycolic acid is a component of Mycobacterium tuberculosis. Let's try one more. Here's one more. Which of the following inhibits cell wall formation by preventing the transport of peptidoglycan disaccharides by bactoprenol? Is that vancomycin, aztreonam, bacitracin, penicillin, or cephalosporin? Right, bacitracin. Great job. So in this video, I've talked about peptidoglycan synthesis. We looked at the role of bactoprenol in transporting NAG and NAM across the plasma membrane. We talked about transpeptidase, which makes a cross-link between the amino acids attached to NAM molecules. And we looked at autolysins as well, which break the bonds between NAG and NAM, as well as breaking the cross-links between adjacent NAM molecules. We also talked about the structure of peptidoglycan. These things can show up on your exam, so you want to be sure that you're familiar with them. We also talked about how beta-lactam antibiotics bind transpeptidase, which is also, by the way, sometimes called penicillin binding protein. So the beta-lactams bind transpeptidase and inactivate it so that it can't cross-link NAM molecules. We described 2 methods that bacteria use to inactivate beta-lactam antibiotics. One of them was the beta-lactamase enzymes that snip the bonds between the atoms of beta-lactam rings, which inactivates them. The second way that bacteria may inactivate beta-lactam antibiotics is by changing the target. So some bacteria carry the genes for mutant transpeptidase enzymes that penicillin is not able to bind, and that's how they get around the drug. We also talked about 4 methods that scientists use to overcome resistance to beta-lactam antibiotics. We looked at, for example, the production of clavulanic acid and packaging that with penicillin drugs. Remember, clavulanic acid binds beta-lactamase enzymes and inactivates them so that the penicillin can do its job. We said another option would be to strip all of the chemistry away from the beta-lactam ring. This is how the monobactam antibiotics are made. These monobactams are not recognized by the beta-lactamase enzymes as easily. We said that some antibiotics, like the carbapenems, change the atoms that are in the penicillin so that the penicillin is not recognized as readily by the beta-lactamase enzymes. And finally, we said that you could change the chemistry around the penicillin, or rather, sorry, let me say that again. You can change the entire chemistry around the beta-lactam ring. This is what your cephalosporins do by adding a different ring that's not in your penicillin nucleus. So even though it's a beta-lactam antibiotic, it doesn't look like penicillin. Two alternatives to beta-lactam antibiotics would be vancomycin and bacitracin. You want to make sure that you know how those work. Those could show up on your exam. You could get a question, for example, that lists a bunch of penicillin drugs and then a let me say that again. You could get a question where you have a bunch of beta-lactam antibiotics listed, penicillin derivatives and cephalosporin, for example, and then in the middle of that have bacitracin or vancomycin, which are not based upon the beta-lactam ring. And you would be asked which one of these doesn't belong. I would hope that you would choose bacitracin or vancomycin because they are polypeptide antibiotics, not beta-lactam antibiotics. We also talked about two antimycobacterial antibiotics. We talked about isoniazid, which inhibits mycolic acid synthesis, and we talked about ethambutol, which inhibits the incorporation of mycolic acid into the cell wall. This was a kind of long video, but all of these topics go together, so hopefully you have a better understanding of how antibiotics can inhibit cell wall synthesis. In the next video, we'll talk about how antibiotics inhibit protein translation. I look forward to seeing you in that video.